Now we can see another vector which is derived from Puck series vector only, Puck vectors only, pGEM3Z. This particular vector pGEM3Z is having, like, all the features are same only, but two additional features are there. Before we go to those details, we can see what's the importance of this particular vector. This vector is quite useful for in vitro transcription work. This is useful for in vitro transcription. So what's the need of in vitro transcription? See, we know in vitro replication is there, like our polymerase chain reaction. If you want to make maximum many copies of your particular piece of DNA, then you can depend on PCR. In the same manner, you can do transcription work and translation work also. Like you can do it in a test tube. So here, this particular vector is designed for in vitro transcription. What is the advantage of doing in vitro transcription? Suppose you want to get the RNA transcript of a particular gene of interest in very high amount. In that case, this is an excellent tool. Why? Because, in the cellular environment, the host chromosome is there, that will also definitely code for lot of RNA species, many number of RNA molecules will be there, and from the plasmid also the RNAs will be there. So like that, it will be a very complex environment only. So from that, if you want to purify your transcript of interest, that will be difficult. But if you do the entire process in a test tube, like here we are using this particular DNA molecule as such for the process of transcription, we will just add the enzyme which is responsible for converting, transcribing your gene of interest into the RNA transcript. It is RNA polymerase enzyme only. RNA polymerase enzyme plus this vector, that will give you RNA transcripts. And here I said, that's one thing. Like if you want to go for probe synthesis, like you want to use this RNA as a probe, then in very high amount, and you can very easily isolate and purify this particular transcript. That is one thing. Second thing is, suppose you want to study the processing, post-transcriptional events and all, like intron splicing and tailing process, like you want to study your RNA in a great detail. In that case also, you can go for in vitro transcription. And the third case is, like, sometimes some gene products like proteins, they are toxic to the cell system. So cell will not survive if you go for producing that particular protein in the cell system. So what we will do? We will do in vitro transcription, and then we will go for in vitro translation. So with in vitro translation, you will be getting the protein product of your interest without the help of a bacterial system. So there is no problem like the cell culture will not exist. So such kind of problems are not arising. So these are the advantages. Like if you want to go for synthesizing probes in RNA probes in very high amount, then you can go for in vitro transcription. Or if you want to study the RNA processing events, then also you can go for in vitro transcription work. And if the protein product that is of of some toxic nature, for the bacterial system, in that case also you can go for using a combination of in vitro transcription and in vitro translation. Now we can see the features. See, everything is like the origin of replication, that always required. Then ampicillin resistant gene, the same gene is there, and then you have lag-Z segment, the same thing is there, and then multiple cloning site, everything is same. Here you will insert your gene of interest, but two additional elements are there. The first thing is your T7 promoter, and the second is sp6 promoter. So now what is this T7 and sp6? T7 and sp6, these are bacteriophages only. And you are keeping the promoter segments of the genes of T7 bacteriophage and sp6 bacteriophage. Promoter segment of the genes present in T7 and sp6. What's the need? This is a bacterial plasmid only. In that bacterial plasmid, you are keeping two bacteriophage promoters. We had already discussed this kind of things, I hope. We know the enzymes of viruses, like bacteriophages are viruses only. This is like T7 promoter that will be recognized only by T7 RNA polymerase enzyme, and sp6 will be recognized by sp6 RNA polymerase. And this T7 RNA polymerase, its rate of synthesis, like transcript synthesis capacity, that is far higher than your bacterial RNA polymerases. Compared to bacterial RNA polymerases, T7 RNA polymerase and sp6 RNA polymerase, their synthesis rate is very high. They can produce RNA transcript in very high amount in short period of time. So as we as genetic engineers, we are always having commercial interest. We see all the aspects like how it becomes economical and all. So when we think about such things, always it is good for us to produce very high amount within short span of time. So if you use a T7 promoter, the sequence which is like coming right after this promoter, that will be transcribed in a very high rate. We can say some 1 to 2 microgram RNA per minute. That's very good amount compared to your RNA polymerase, bacterial RNA polymerase. So you will be getting expression of this particular gene at very high rate. You will be getting lot of RNA, 1 to 2 microgram RNA per minute. But there is one question now, like T7 promoter and sp6 promoter, for what you are keeping 2 promoters. We know for a gene, particular gene, there is 1 promoter. But here you are keeping 2 promoters, and that too in opposite orientation. This is very, very, very important. As you are keeping 2 promoters in opposite orientation, the advantage is both the strands can act as template. You know, in the transcription process, only one strand is acting as a template. In replication, both the strands are acting as templates. But here, only one is acting as template. Just by keeping 2 promoters in opposite orientation, you are getting, like you can go for getting the transcript of both these strands. But at that time, you will go for using either T7 RNA polymerase or sp6 RNA polymerase. If you put T7 RNA polymerase, then T7 promoter only will be get recognized, and accordingly the strand will get synthesized, will be copied. And sp6 if you use, then the strand which is only one, the other strand will be getting transcribed. So like that you have 2 promoters. 2 promoters ensures that your both the strands are getting transcribed, and according to your interest you can go for selecting the RNA groups. So this is the advantage of in vitro transcription, and the vector system which is used for this work is pGEM3Z.